but I'll try to talk a little more loudly so that everybody understands. So as my title suggests, I'm going to talk about a systems-based approach that I developed in Richard Maas's lab during my postdoc years, which we have used successfully to discover new genes which are important in the ocular lens development in mammals. And by the end of the talk, I would, mid through the talk, I would talk about one gene which is called as Tdrd7, which we identified, which is a novel cataract gene, a bit about its molecular function in the lens, which has opened up new doors. And then to conclude, I will talk about my future directions. So the eye in vertebrates is a complex multi-component organ. So roughly, it can be separated into three different parts: the anterior part, which contains the cornea; the posterior part, which contains the retina and the optic nerve; and the intermediate part, which contains the lens out here. And the lens is a unique component of the vertebrate body because it's a three-dimensional component. It's a three-dimensional tissue which is completely transparent, as can be seen in this picture out here. And why should we be thinking about our lens is because as we age, there is a good chance that we would end up getting a cataractous lens, which means that there would be an opacity in our lens. And this is what an individual with a cataractous lens sees the world. And it remains the leading cause of blindness, affecting 77 million individuals worldwide. In US alone, there are 3.1 million cataract surgeries which are performed annually, which roughly translates into the most expensive surgical expense amounting to $3.4 billion, roughly over 2.5% of the Medicare budget. And the sad part is that between 10 to 25 estimated ocular cataract surgeries develop complications wherein one has to go back and remove some of these opacities which are left behind because of epithelial to mesenchymal transitions. So any attempt at understanding how the lens forms and how it maintains its transparency will help us to develop strategies for prolonging transparency in the human lens. So let me begin by introducing how lens induction takes place in mammals. It begins shown here are stages from E9.5 mouse embryonic time points, and shown here in orange is the optic vesicle, which interacts with the surface ectoderm to cause a thickening called as the lens placode. And these two structures coordinately develop by which the surface ectoderm pinches off to form the lens vesicle, is seen as a spherical structure here. And this lens vesicle is eventually filled up by the posterior part of the lens, which are posterior fiber cells, and in the anterior part, it consists of anterior epithelial cells. And these anterior epithelial cells, throughout the life of the organism, continually exit the cell cycle in this zone of transition and begin differentiation into what are called as secondary fiber cells. And these are sort of piled on as in a sort of a structure as an onion to form this lens. The important part to note here is that these stages which occur in development of fiber cells, the anterior epithelial cells differentiating into fiber cells, occur throughout the length of the life of the organism. So the principles that we learn for how these fiber cells are differentiated can be applied throughout the life of the lens. So as a lens biologist, I would like to believe that lens cells are unique, lens fiber cells are unique. And why I say unique is because when an epithelial cell exits the cell cycle, it undergoes a dramatic change in cellular physiology. And what I mean by dramatic change is that it has to upregulate genes which are expressed in a fiber cell specific manner, and it has to downregulate genes which are expressed in the anterior epithelium in this layer of the cells. And we know that if we misexpress genes which are important in the anterior epithelium, even in these second or third of the fiber cells out here, it disrupts the entire differentiation program. So it's very critical that these transcripts are degraded, these proteins are not made beyond this zone of transition, and that eventually these cells are making really essentially copious amounts of crystalline proteins. And again, high levels of crystallines, by really high and copious levels, I mean this structure accumulates around 450 milligrams per ml of crystalline proteins by the time that it is complete, the differentiation is complete. The other challenge that the lens fiber cell has is that as it's differentiating, it also has to elongate and fill up this vesicle, and it has to make all these proteins before, make all the transcripts and then translate them into the proteins before all of its nuclei is degraded, all of its subcellular components are degraded, and it ends up being a massive bag of crystalline proteins. So needless to say, when these processes are hampered with, you end up getting an opacity which is termed as cataract. So how really unique are fiber cells? If you talk about elongated cells, we have examples of neurons which are elongated. If we talk about cells which are on a timeline to produce transcripts and then translate them into proteins, we have the spermatogenesis pathway, wherein although the differentiating sperm do not lose their nuclei, they undergo such high level of chromatin compaction that they lose ability to transcribe mRNA. So they have to have a way of really storing transcripts for future rounds of translation. And when we talk about polarized cells, then we have examples of migrating fibroblasts where cells do have polarized ends towards where they are migrating. And all of these cell types are united by a subcellular component called as the RNA granule. And it has different names in different cell types. As we understand more about it, I think we will be able to define them much better. But in the neurons, they are called as neuronal granules. In these differentiating sperms, they are called as chromatid bodies, and in these differentiating fibroblasts, they are called as transport granules, which physically transport mRNA from the nucleus onto the leading edge of the fiber cell. So for the purpose of today's talk, when I talk about post-transcriptional regulation, post-transcriptional regulation can occur on a life of a transcript as it is transcribed from the DNA and from being spliced, edited, exported into the cytoplasm, where it can it undergoes stability, localization, and eventually translation. And through all these stages, the parts that I would highlight when I talk about post-transcriptional regulation are the cytoplasmic aspects of it. So let me step back and really tell you how it all started. When I joined Richard Maas's lab a few years ago, I had 3 specific aims, my own personal aims. I wanted to be able to really make a tool by which I could predict genes which have a function in lens development. And then I wanted to predict these, test these predictions in human patients which had pediatric cataract. And in the event that we found a novel gene, I wanted to use animal models to be able to gain insight into the function of these candidate genes. So the tool that I have generated is called as iSyTE. The I stands for integrated, SyTE stands for systems-based tool for eye gene discovery. And it is based on a very straightforward principle. We wanted to know what is present in which transcripts are expressed in critical stages of lens development. But that's not all. We also wanted to be able to assign what we call as enrichment p-values to each of these transcripts which are expressed in these critical stages. So what we did was we identified 3 stages, that one being from the E10.5 mouse lens, where it invaginates from the surface ectoderm, through E12.5, where primary fiber cell differentiation has initiated. And we dissected around, on an average of 600 lenses per biological replicate sample. We did this in triplicate, and we generated some extremely high quality microdata sets, which were for each of these lens sets. But the next part is what is interesting. Now we could look at each of these data sets and we could say, okay, these are the transcripts which are present in this particular stage of the lens. but we would not be able to comment on how exclusively these transcripts are present in this particular stage. To generate a p-body for the exclusivity of expression, what we did was we used a whole embryonic body from each of these time points, removed the ocular tissues, and we generated, again, very high quality microarray data sets for these whole embryonic time points. And this we used as a reference data set. And then what we did was we carried out a Bayesian t-statistic comparison between these two data sets. And an analogous experiment would be an in silico subtraction, a cDNA subtraction that we were used to as molecular biologists when we were looking at two different cell types and we wanted to know cell type specific genes. So this is analogous to that. We call it as in silico subtraction here. And this allowed us to generate p-values which are called as lens-enrichment p-values. So how effective is iSyTE? If you use this data set, if you look at data sets from each of these lens time points and you arrange them as the top 100 genes based on detection p-values, and you ask the question, which of these genes can I recognize through my knowledge of literature as genes which are involved in lens development, we get 0 out of the top 100 candidates to have a known role in lens development. So non-subtracted list, 0 out of top 100. If you do this subtraction, you get almost 50% of the top 100 genes have a known role in lens development, which leaves another 50 genes which would be really important, again, scattered through this list of 100 genes that we had to look carefully into. Another way of really testing whether iSyTE is working efficiently is to look at the existing knowledge of the lens induction pathway. And this was generated by a careful really survey of the literature, and each of these node is really accounted for by a published report. And as a lens community, we depended, we could generate this. So I generated this gene regulatory network, but it is based on really 20 years of hard work from individuals who worked on the lens. And as a lens community, we depended on orthologs found by Drosophila researchers. We depended on a mouse geneticist who had mapped a cataract mutation, or we depended on human patients with pediatric cataract that we could solve and assign genes for mutations. So this was a painful way, and of course, a lot of mouse genetics, after which we could assemble a rudimentary, this is what I call a rudimentary gene regulator network for just the stage of lens, presumptive lens epithelium going into a lens pit formation stage. So had we known, had we got access to iSyTE, what we would have been able to do is identify a vast majority of these nodes a priori. And again, we would have to do our mouse genetics and other experiments, benchworks, to be able to establish these nodes, establish these edges between these nodes. But having known majority of the nodes would have been a great advantage. One more way really to ask the question is, could iSyTE really enable us to find genes which are already associated with cataract? And the quick answer to that is that when we looked at the top 25 non-syndromic cataract genes which are mutated in non-syndromic cataract cases, and we asked the question that had we not known this gene before, had we just looked at the original interval, mapped interval where this unknown gene was, could iSyTE predict it? And the answer is that if you were willing to sequence the top 5 candidates in a particular interval, which could have anywhere ranging between 50 to 100 genes in that particular interval, iSyTE could find 90% of these candidates within the top 5 genes that it basically could give as an output. Another way to really look at it is through this database that we have generated now, which would be going online as this publication is going to be accepted. This would be online as a UCSC genome browser tracks sites. And the way it would work is that we have generated a way to convert these p-values into a color code. And so the most significant lens-enriched p-values are coded by the brightest red color. So this is an actual gene, GJ3 or connexin 46, which was predicted in a 5 MB region from from an interval mapped for pediatric cataracts. And one can see that these bright red color tracks using iSyTE are the ones which are delineating GJA3 and recognizing GJA3. So if you were a human geneticist working on cataracts, all one has to do is go online on the UCSC browser, turn on our iSyTE tracks, and just put your interval region on there, and you could see whether a particular gene was popping up as the brightest red color gene, which was the gene that you would go, or the set of genes that you would go after and prioritize to find the mutation. So this is another example to show that crystalline genes are, this is a gamma crystalline cluster, and you can see that they are enriched. These three tracks are for the 10, 11, and 12 different time points of the lens, and one can see that it gets progressively enriched, and so it could have been found in a 15 MB interval. And these are not just limited to just crystallines or structural proteins which are highly expressed in the lens. One could have also found PTEX3, which is a transcription factor, by this approach through a 3 MB interval region. And the biggest one of them all is really PAX6. This is really an important gene in lens development, and we could have looked at a 30 MB interval region and also predicted PAX6 to be an important gene in lens development. And I would like to dwell one more slide on PAX6. PAX6 is this candidate gene which, if you misexpress a human PAX6 into Drosophila, it gives you a complete compound eye. This is a picture taken from a seminal report from Walter Gering's lab back in 1995, which was one of my inspirations to start working on lens, really, and PAX6 for that matter. We know that the converse experiment, you take the eyeless, the PAX6 homologue from the Drosophila and misexpress it into Xenopus head ectoderm, can also give rise to complete eye ocular tissues. So iSyTE could enable us to find really critical regulators of lens development. So now that we were confident that we had formulated a tool for predicting genes which are important in lens development, we wanted to use it in map cases of cataract. And I decided to participate in a project initiated by Dr. Cynthia Morton at BWH. This project is called Developmental Genome Anatomy Project, and or DGAP. And the principle of it is that if we could identify patients which had developmental disorders and who carried balanced translocations, if we could nail down the genes which are affected as a result of these breakpoints in their balanced translocations, we should be able to assign the gene to the particular phenotype that we were interested in. So as part of DGAP, we recruited a young male who at age 2 had developed bilateral cataracts, severe cataracts, as can be seen in this picture right here. And where the breakpoint was located on chromosome 9, there was an inversion, and it was a 5 MB interval with 50 candidate genes in that interval. So a perfect setting to really ask the question whether iSyTE would be able to predict a new gene for us in this particular patient. And indeed, iSyTE predicted that a gene called as Tdrd7 was the most likely candidate in this particular interval of 50 genes. What is Tdrd7? Tdrd7 gets its name from Tudor domain containing protein containing 7. It's a family of genes actually having 10 family members. Tudor domain proteins are conserved from Drosophila to humans, mostly found in the context of germline development. And Tdrd7 is unique because it's only one of the 2 Tdrd family members in humans which has got this interesting 3 repeats of what is known as the OST-HTH domains. These are predicted to bind RNA, while it gets its name from these 5 Tudor domains at the N-terminal, which are predicted to bind methylated arginines. And in all contexts that Tudor domain proteins have been found to date, they have been implicated in some or the other aspect of RNA metabolism. So coming back to our patient, was our prediction right? Tdrd7, was it really broken in DGAP 186, or the patient with cataract? And we performed fluorescent in situ hybridization analysis. This is a reference to show that there is a reference probe in green and Tdrd7 specific probe in red. And if truly there is an inversion in the patient as we have predicted in Tdrd7, the Tdrd7 signal would be split into two, as can be seen in this particular picture out here, that the signal is split here and away from the chromosome, internal part of the chromosome. In here, in the normal equivalent chromosome, you don't see that particular split. This was further confirmed by Southern blot analysis, where we predicted aberrant band by TAC1 or STU1 digest only in the patient samples, as can be seen here. And further analysis refined this breakpoint to be lying between intron 2 and 4. And the direct prediction of that was that the one copy of the Tdrd7 would be broken and would rise to haploinsufficiency in this patient. And we tested for haploinsufficiency in this patient lymphoblastoid cell lines on both the RNA and protein level by quantitative RT-PCR analysis and Western blot analysis, and we found that indeed that was the case, that Tdrd7 dosage was reduced in this patient with cataract. So armed with this information, we decided to go out in the world, seek out collaborations with human geneticists and with individuals who had been mapped, who had congenital or pediatric cataract in their families, and where the mutation had been mapped on chromosome 9. We wanted to seek out whether it did have a mutation in Tdrd7, and a second independent case of Tdrd7 mutation resulting into cataract. So we found one such family in Kuwait City, where there were 4 individuals which had inherited a mutation in Tdrd7 open reading frame. It was a 3 base pair specific deletion which had been inherited by only those individuals which had developed congenital cataract. And what this 3 base pair deletion did was it removed a valin which has been conserved from humans to zebrafish, and which we predicted would affect the first Tudor domain of the Tdrd7 protein in these individuals. So we now were confident that we were on the right track. Tdrd7 was indeed perhaps one of the new cases of cataract genes predicted by iSyTE in humans. So we decided to go forth ahead with our animal studies to find out gain insight into Tdrd7 function. The first thing we did was perform section in situ analysis. This is a Tdrd7 specific probe. This is a section of a E12.5 mouse lens, and one can appreciate that Tdrd7 expression is localized at this stage very specifically to the ocular region, as can be seen here. On closer inspection, one can see that Tdrd7 expression is restricted to the posterior part of the lens in the fiber cell compartment. This is the anterior part, and this is the retina. This is the lens. And more importantly, that this expression was conserved in chicks as well as in mice, as can be seen here. So the chick opened up a new door for us while we were generating our mouse mutants. And what we could do was we could assess rapidly whether Tdrd7 was indeed the right gene to go after. And what we did first was we used a retro virus construct which had been labeled with, which had a GFP construct in it, and we targeted chick lenses injected at stage 10 or 11 chick embryos, and we found that we could target the lens very specifically in over 80% of these animals. So once we could do that, we used this as a vehicle to deliver shRNAs targeted against Tdrd7, the chick Tdrd7, and we found, I'm hoping that this is a little clear, but one can see these opacities here, one can see that, and it's slightly more clear in my monitor out here. But we could essentially recapitulate the Tdrd7 mediated cataract phenotype in chick embryos. So then we decided, we were confident that we were on the right track. This was the gene to go after and invest in the heavily costly mouse studies. The first thing we did was we generated an antibody against the mouse Tdrd7. Shown here is an expression of Tdrd7 at an E12.5 lens, mouse lens sections. Here the AEL is on the top. This is oriented slightly differently from the previous picture, the lens that is. And one can appreciate that Tdrd7 is expressed in the cytoplasm, and on closer inspection one can see that it's expressed as granules and can be seen in perinuclear fashion as well as restricted on the anterior part and the posterior part of the lens. So since Tdrd7 or Tudor domain proteins were implicated in RNA metabolism in previous studies, we decided to stain the E12.5 lens with cytoRNA select dye available commercially from in vitro gen, which preferentially binds RNA. And we asked the question whether these granules had elevated amounts of RNA, cytoplasmic RNA, aggregates of cytoplasmic RNA in them. And we could see that indeed, I hope this is visible. You guys have to tell me because it's not from this angle that I'm seeing. But there are these spots out here which co-localize beautifully with these Tdrd7 granules. And then we use another RNA, preferentially RNA binding dye called pyronin Y, which maybe is much more clearer here, which was also co-localized at the E12.5 lens. So we were confident that Tdrd7 granules had aggregates of RNA in the E12.5 fiber cell cytoplasm. So let me dwell a couple of slides on RNA granules again. There are 4 different kinds of RNA granules found in metazoan cells. Upon stress, mammalian cells can assemble these cytoplasmic bodies called as stress granules, which can be seen here. And these granules are sites of really post-transcriptional regulation where the heat shock proteins and molecular chaperones encoding transcripts are selectively left out, and all the housekeeping genes are stored for some time or either channeled for degradation in a second related structure called as P bodies. But unlike stress granules, P bodies can be found in endogenous cells as well. P bodies are site of mRNA triage. They have an XRN mediated decay factors. They have decapping enzymes. They are small subcellular machines which can degrade RNA. And all of these really granules are being looked into very much in detail since the last really 10 or 15 years. So it's very recent work really. I would say really 10 years mostly in the molecular level. Then there are chromatid bodies which are found in differentiating sperms, which are again implicated in post-transcriptional regulation during that process, and transport granules, as I talked before, in neurons as well, where, for example, stophan is a molecule which can transport mRNAs to the sites of the synapse where you need protein translation at that particular site when you are making that particular synapse. So in an elongated cell, transport of mRNA from one part to the other part is highly critical. So there are these 4 different kinds of RNA granules which are roughly classified into these classes. This is still a work in progress. As cell biologists define better markers, we would be able to better classify these granules and differentiate between them. But as lens biologists, we could foresee that a lens which is under these heavy demands of making high levels of proteins while degrading other transcripts, and also one thing I forgot to mention was crystalline transcripts are really high in numbers versus that of transcription factors like PROX1, which are also expressed in the fiber cell compartment. So how do we get these really differences? You still end up making a good amount of PROX1 protein. You end up making a lot of crystalline proteins. So how does the cell differentiate between these vastly differing amount of transcripts? So we thought that RNA granules could be entities, cellular entities, which could help stabilize these particular transcripts in the fiber cell cytoplasm. We could also foresee perhaps that there are stophan RNPs in fiber cell compartment, which may be important for transport of RNAs, or stophan mediated decay in the fiber cell compartment, or P bodies, which are generally important in translational arrest and transcript degradation. So long story short, we looked at a vast array of markers, and we found that stophan 1, which is ortholog of the Drosophila stophan. In Drosophila, stophan is important for physically transporting the Oscar mRNA to a pole and then generating a gradient of the Oscar protein from that particular pole. And we found that in the polarized lens cell, stophan 1 is co-localizing with Tdrd7 in the anterior edge of these fiber cells, which are elongating towards the anterior epithelium to fill up this vesicle. And one can see this bar graph represents 1 micrometer thickness, so one can see that this localization is intricate. So then we decided to do one experiment in lens epithelial cell line. We decided to see that if we knock down Tdrd7 and we force the cell to make stress granules or elevated number of P bodies, if we downregulate Tdrd7, what would be the effect on these cells? And we found that downregulation of Tdrd7 actually had a hampered stress response, as can be seen here, in 2 different lens epithelial cell lines from humans as well as mouse. So before I go ahead, let me summarize what we have learned up till now. We've shown that Tdrd7 expression is highly lens-enriched in development. We've shown that Tdrd7 knockdown in chicken lenses results in cataracts. We've shown that Tdrd7 localizes to cytoplasmic RNA granules in fiber cells, and that these granules also co-localize with stophan 1 RNPs in fiber cells, and that they might be important in the regulation of a stress response. So to gain further insight into this mechanism of Tdrd7 action, we decided to perform experiments in a mouse knockout model of Tdrd7. And before I go ahead, I want to just give the result upfront. When we knock out Tdrd7, when we generate a Tdrd7 null mutant in mice, what happens is pretty dramatic. What one can see here is you can see the pupil here, but this is the anterior chamber of the eye. It's looking at a mouse eye right from the front. And what has happened is the lens has disrupted the posterior part of the capsule, managed to float from the back of the eye to the front of the eye and enter into the anterior chamber, and this is the fibrous lens sort of floating around in the anterior part of the mouse eye. This is not supposed to happen really. So it's a very dramatic phenotype when Tdrd7 deficiency is generated in mice. So really this mouse was a result of a collaboration, a very fruitful collaboration from Dr. Simon John's lab in Jackson Labs. And he was interested in screening for ocular mutations based on an ENU screen. And when we teamed up together, he had an ocular phenotype which was dramatic, as I just showed you. And we decided to look into Tdrd7. Lo and behold, that mutation was mapped to the Tdrd7 locus in the mouse. And we found that there was a specific cytosine to thymine change in the 2187 position in the Tdrd7 open reading frame. What this did was generated a stop codon in the frame, and the direct prediction of this was that you would have a missense mutation and there would be no Tdrd7 protein formed in Tdrd7 null zygous mice. And indeed that was found to be the case. When we looked at Western blot analysis from the Tdrd7 null mice, the ocular regions here, or in the lenses with immunofluorescence, we found that there was no Tdrd7 protein expressed in these mice, which were a Tdrd7 null. So indeed, that particular mutation generated a null allele. So some more characterization of the mouse phenotype at around 1 month stage. One can already see with a slit lamp exam that one can detect a cataract right here. I'm hoping that it's visible here. But by 1 month of stage, as I showed you, this is a histology of the mouse ocular region. And one can see that there are these intense vacuoles formed, and this fibrous material of the lens is disrupting the posterior part of the lens. Eventually, this is the material which will find its way out and start floating in the anterior chamber, as can be seen here. By 3 months' time, you can see that the lens is pretty done cataractous by this time. So it's a very dramatic phenotype, 100% penetrance. So we decided to rapidly generate a strategy to identify genes which are regulated by a cataract and to get into the molecular mechanisms regulated by Tdrd7 and get an insight into the molecular mechanism by which Tdrd7 regulates lens development. So we decided to take a microarray based approach, a gene profiling based approach, and we decided to scheme it as the following. One, we decided to look at mouse lens epithelial cell lines which had Tdrd7 knockdowns. These cell lines only partially recapitulate the lens cells physiologically. So it would be a very stringent test. Only the core regulatory events could be found using this particular cell line. But nonetheless, we decided to use this in our screen because we wanted to find the most meaningful candidates. The second microarray analysis would be done on lenses of Tdrd7 null mouse mutants at P4 3 weeks before the cataract was generated, and at P30 after the at the point of where cataract was detected. So we wanted to compare differentially regulated genes between these different data sets across the board, and through a literature curation step, come up with a list of most promising candidates that we wanted to then focus on to find this molecular mechanism. So to my surprise, actually, to my personal surprise really, I thought the Tdrd7 knockdown mouse lens epithelial cell line may not work as well, but it actually gave us a lot of clues. When we looked at Tdrd7 knockdown and looked at which genes are differentially regulated, we found that there are genes which are important for granule formation like G3BP or HSPB1 which were downregulated. We found that P-bodies, genes very important for P-bodies like DDX6 were downregulated, or some fiber cell specific dead box helicases were downregulated. We found that genes which are important for proper lens differentiation like gamma crystalline, EPH-A2, PROX1, SOX2 were all downregulated. And we also found that interestingly, PAC6, E2F, PCNA, and JAG1, which were actually genes expressed in the anterior epithelium, were upregulated. So somehow those were the level of repression was elevated from those. So interestingly, when we compared this with the lens mutants, we found that 6 of these genes were actually even commonly found to be downregulated in the Tdrd7 mutant mouse lenses too. So these represent likely candidates which are more dramatically affected in the mouse lenses. So this is a comparison of the P4 and the P30 mouse lens, and one can see that by P30, a range of crystalline encoding transcripts are downregulated, and multiple genes which, when you have mutations in either mouse or humans, give to cataract or lens disruption of lens developments are downregulated. And also there are genes which are important for granule formations and stress response like HSPB1 which are downregulated. So then we decided to focus on a few candidate genes from these multiple approaches, and we focused on HSPB1 because it was a gene that was found through all 3 screens. HSPB1 is a heat shock protein. It's also known as HSP27, and it interacts, physically interacts with crystalline alpha A protein or crystalline alpha B proteins, both of which, when you have destabilizing mutations, can give rise to cataract. EPH-A2 is a gene which is downregulated in Tdrd7 null mutants, and there is an interaction of EPH-A2 and HSPB1. Sparc is another gene which is downregulated in Tdrd7 null mutants, and so is crystalline beta B3, all of which are known to be important for lens development. So this is where really I think science gets really interesting and fantastic, because I looked up the literature for Sparc and EPH-A2, and I found that in 1998 there was a mouse mutant made for Sparc, the Sparc gene, and we found that the histology of this particular mutation had the same phenotype, vacuoles, as well as a dramatic rupture of the posterior capsule in the Sparc1 null mutants, but this occurred at 3 to 6 months of age. And the same rupture of the posterior part could be seen in EPH-A2 null mutants too, but at 8th month stage. So we think that the reason the Tdrd7 mutant is more severe at an earlier stage can also be, of course, because of strain differences But one argument can be made that it does control a vast number of genes which are important for lens difference, lens formations, which is why it's got a much more severe phenotype. So to then ask the question whether any of these candidates represented direct targets of Tdrd7, we performed an RNA immunoprecipitation assay using the Tdrd7 antibody, and we asked the question whether any of these candidate genes could be pulled down by the Tdrd7 antibody. And we found that crystalline beta B3 and HSPB1 could be pulled down by the Tdrd7 antibody out here. But to my surprise, EphA2 and Sparc, which I thought were really the prime candidates for a direct sort of regulation of Tdrd7, were not as enriched as these two candidates out here. But then again, when we go back to the literature and we find that there has been a study where Stauffen-1 RIP experiments have been done, so RNA immunoprecipitation with the Stauffen-1 antibody, and they have asked the question which of these proteins can be enriched in by the Stauffen-1 pull down, and EphA2 and Sparc are transcripts which are pulled down by the Stauffen-1 antibody. So this allows us to generate an elegant model where we think that Tdrd7 is protein is directly binding to or in a complex more directly binding to beta B3 crystalline and HSPB1 transcripts, or through its interaction with Stauffen-1 in the lens fiber cells, which interacts with Sparc and EphA2, may be able to stabilize these mRNAs and control their translation to maintain lens transparency. Now, this part of the really the model has to be tested in future studies. So to summarize, we have developed iSyTE to facilitate cataract gene discovery, identified a novel cataract gene Tdrd7, identified RNA granules in the developing lens, characterized the Tdrd7 null mutant mouse model of juvenile cataract in humans. We've shown that Tdrd7 regulates lens-specific genes either directly or via interaction with Stauffen RNPs. And perhaps for me, as a person who studies transcription factors and still studies transcription factors, the biggest take-home for me personally was that I used to think, like we think about tissue-specific transcription factors, now I have to start thinking of tissue-specific components of the post-transcriptional regulatory machinery, which may be functioning in mammalian organogenesis. So in future directions, independently in my own lab, what I would like to do is really expand this analysis of Tdrd7 mRNA targets to really look into chick lenses, because they are bigger lenses, and to try to see the whole repertoire of Tdrd7 transcript targets. I would also like to use the Tdrd7 specific antibody, which is really working well, to find out genes which are important for which are interacting with Tdrd7 in the lens. And again, this approach has been very successful in other Tudor domain proteins, where they have started generating an interactome for that particular Tudor domain protein. So we would like to also start putting together an interactome of Tdrd7 in the lens. And we have already started doing some co-IPs with some specific proteins which are likely candidates, which have been found by iSyTE as well to be important in lens. And we would also like to pursue whether Tdrd7 has any other distinct post-transcriptional regulation events in the lens. And by that, I mean that in a newborn lens, we also see this curious phenomenon where one can still see all these granules in these differentiating fiber cells. But as one gets into this zone before the nuclei are completely degraded, one can see that Tdrd7 also is expressed in the nucleus and then comes out on the surface of the nucleus. If you look at the confocal 3D picture of this reconstruction of this nuclei, one sees that this Tdrd7 is actually coming out on the surface of that nucleus, and then a few cell layers later, the nucleus is completely degraded. So that's a complete mystery to us right now, what it is doing in this nucleus at this particular stage. That's something that we would like to look into. And then we have some inclination to believe that Tdrd7 is also functioning in other tissues. For example, we find that in 2 of the patients who develop congenital cataract, they also develop glaucoma. And in our mouse mutant model, we find that there was elevated intraocular pressure in these Tdrd7 null mutants. And one could see that the ganglion cell layer was also completely diminished in these Tdrd7 null mutant mice. This is a hallmark of glaucoma. So whether this was due to a secondary effect of the lens being disrupted, or whether Tdrd7 has an indigenous function in the retina or in these drainage structures, can be addressed when we have a Tdrd7 conditional null mutant, which we would address in future. The other interesting fact is that, as I spoke before, there are these chromatid bodies which are found in the mouse spermatogenesis pathway. And we find that in Tdrd7 null mutants, there are no elongating sperms in these testes of Tdrd7 null mutants. And we find that Tdrd7 is a component of the chromatid bodies in these heads, but in nulls, obviously there is no Tdrd7 protein. We have some evidence that these chromatid bodies are also not normal, but we are gaining more evidence to show that by staining of other granule components, that these granules are definitely defective in these Tdrd7 null testes. So this is a area that we'll have to we'll start looking into. And there are different kinds of RNA granules. I sort of glossed over it in my talk, but we have found that there are P bodies in the lens, high levels of P bodies. Now I want to remind you that in this structure of the lens, at this stage, there is a high level of protein transcript formation and translation going on, but in crystalline, for the crystalline proteins. But yet one can see a large number of P bodies in this compartment. And as I said, P bodies are associated with transcript degradation and silencing. So how is this compartmentalized? One simple hypothesis is that you have Tdrd7 components which are taking care of the genes which are important in the lens fiber cell compartment, and then you have these P bodies which are degrading all the other mRNAs which are not required in that particular compartment. But how does this specificity arise? This is one direction we would like to take. I just want to mention that these are not just spots that we see in the lens. We actually can administer cyclohexamide to these E12.5 carrying females, and 4 hours later when we look at the lens, all of these P bodies are completely dissolved, indicating that they are bona fide P bodies found in the lens. The other thing is that as we look at later stages of the lens, we find that P bodies are restricted in the anterior epithelium out here, as one can see. And if I could remind you, Tdrd7 was expressed in the posterior part of the lens. So not only there are RNA granules in the lens, they are located in different parts of the lens, in different cells. And we have received some funding for characterizing these P bodies and the transcripts which are associated by these P bodies in the anterior epithelium of the lens. This is in collaboration with Dr. David Beebe at WashU. This grant had just started this month, actually. So another future direction is that I have collaborations going on in the Middle East, in the middle of Iowa, and really in the middle of Colorado as well. And I have a cohort of genes which has been prioritized by iSyTE as candidate genes that may function in lens development. Some of which are these, some of them have already been mapped to loci in humans or mice. These are section in situs. There are all, most of these are genes which have never been described in any context before. So we would be collaborating and pursuing our collaborations, ongoing collaborations with these folks to find new cataract genes. One of them is already in revision. It's a PVRL3. It's a cell adhesion molecule that we have found a patient which has got, again, haploinsufficiency of PVRL3. It's a very specifically anterior epithelial localized molecule. We have found that its interactome is largely present in the lens using iSyTE, and it's another candidate for a new cataract gene. The other approach is to look at mouse mutants, and Dr. Simon John at Jackson Lab is actually given six more mouse mutants which have been independently isolated, and I'm hoping to find the right gene by next week which gives rise to this dramatic phenotype. Again, I want to remind everybody that these phenotypes are not normal. You don't really get these dramatic phenotypes where the lens actually gets disrupted so much and then starts floating in the anterior chamber. This is one of them. So, and it's not mapped to the same chromosomal location as Tdrd7. So potentially there is another candidate out here. Perhaps I would like to think in the same pathway as Tdrd7, but we'll find out. So these are the directions that we have. One more is, again, we have another protein called Zb0b8b that we have found. It's got a very highly specific expression in the anterior epithelium, as can be seen by this section in situ. We have generated a high quality antibody for this. We have generated some experiments with small I mutants, and we have some evidence to believe that Zb0b8 may be upstream of MEIS as well as PAC6, which are really upstream regulators in lens development. So potentially there is really new ocular development factor that we have found using iSyTE. Again, we have established nice mouse lens epithelial cell lines. So a lot of these, this research can be done in cell lines to gain insight into the function of these proteins, and then can be transferred to mice or fish or other animal systems. And this I'm going to just briefly mention that sometime back I had put together a review, and I had proposed that we can, as biologists, when we really put together these data sets using certain specific protocols that I have outlined out here, we should be able to assemble what we call as the oculome, which means that the gene regulator networks for different components of the eye. This is something that hopefully by the time I retire, if I see this happen, I would be really happy. So yeah, so to conclude, I want to thank Dr. Richard Maas, my postdoc advisor, who has been fantastic. He has supported me for all these years, and given me the freedom to write my grants and really pursue my projects the way I wanted to, take them to the directions I wanted to. And again, I couldn't have done it without the initial support that I got from Dr. David Saul, who was my PhD mentor at the University of Iowa, and I used to work on Candida albicans phenotypic switching, which was a really hot area, which still is a hot area in Candida albicans biology. And when I left it in 2003, David gave me the confidence that I could really jump into any system and start working in it. I also want to thank Dr. Simon John, who has been, again, a fantastic collaborator, and I hope to keep collaborating with him in future. Dr. Paul Anderson, who is the world expert on stress granules, and who was conveniently located in Brigham and Women's Hospital, so I could really get his insights into granule biology. Dr. David Beebe has been phenomenal, because although I have never formally trained with him, he has just been an amazing mentor to me. Amazing, amazing. I met the right people, really. And he's at WashU, and he was, being such senior person, he still was generous enough to collaborate with me and write an R01 together, which I would never forget in my life. Then Cynthia Morton, who again allowed me to participate in the DGAP project at BWH. Shamil Sinevev and Gregory Krukev from Broad and Harvard are my collaborators for the iSyTE tool. Fauzan Al-Kuraya, Abdul Mitabid Bebehani, Dr. Arlene Drack, and Dr. Ayn Sayar, 4 ophthalmologists / human geneticists who I have active collaborations with. Dr. Sung Cho and Dr. Martha Bulick. Anton Abu Khalil and Joshua Ho are my really bioinformatician gurus, and who I can go as a biologist and tell them what I'm thinking, and they can start talking into mathematics and get it to work. So I've been really fortunate to keep these things going, and I hope that we should be able to work together for many years to come. So with that, thank you very much for your attention. I appreciate your time.